So I'm hoping you can see this presentation now.
Yes, sir, we can.
Yes, sir, we can see.
Okay, good. So this is what we were discussing yesterday, a little bit of this. And I had said that, this paper is essentially um, one of the contributors to this paper has been a student from DBEB, who was my student, who did MTech with me. And all the computational work he had learned was very useful, I think, in this project that he did with Ron Wise. I hope you can hear me. All right. So, so what you're seeing here is a synthetic circuit which has been done, and we'll talk more about synthetic biology and synthetic circuits later when we come to the specific topic. Here we are looking at, there will be some overlap obviously. Here we are looking at essentially the circuitry in terms of transcription and how that works out, but we can't avoid looking at the logic gates that arise. And so this is like an introductory to the logic gate ideas, but which will be dealt with in more detail in a later chapter, so or a later set of slides. So what we're talking about here is, if you look at this, so this R1 that is here will be synthesizing. This is R1 or R2? It's R2, I think, the way it is looking. So the promoter site is P1, and then this R2 gives you an I2, and then I2 works on promoter site 2. That gives you an R3, then the R3 will go to another site and then works out. You have two reporter proteins. One is the cyan fluorescence protein, the other one is the yellow fluorescence protein. Now, if you look at this circuitry, then this circuitry essentially has first an inverter right there. Inverter means if any signal that comes in here, it will be reversed when it goes out there. And the reason why you're saying this is going to be reversed is because of the repressor. So if there was an up signal here, there would be a down signal there, right? That's the meaning of this. Then this repressor will again repress P2. So that's shown here. But P2 also depends upon another signal, which is I2. So when I2 works on this signal here, it will give you the cyan fluorescence protein, which is R3. Then R3 will then actually directly work on yellow fluorescence protein, but this is an inverted signal again, because if R3 is high, you have a low yellow fluorescence, and if R3 is low, you get a high. So this is essentially the idea that here you have an inverter, here you have a NAND gate. That means if not A, not I2 will give you the signal, right? So together, this is not an AND signal. So the signaling, the truth table, we can look at this later on when we come to that chapter. But here, what is it showing you is that so this one is a very standard system that is used, and I'm saying the standard system is like this: that you have a TET protein which is there, a receptor which will, and then a lac protein which is being produced, which is going to give the inducer 1. This TET will depend upon a signal that comes as anhydrous tetracycline, and that kind of will be working on this R2. So that relieves the stress from there, and then that gives you the synthesis of R3, right? Now, what you see here is the separation. If you looked at this last part of the signaling here, which is this one, so this is an inversion signal that is there. So if this is if you had an ITC here, and that was being used for this whole signaling, then this part of the signaling is what you're looking at. So the response that you see there, if you had a high input, then you can look at this range here, the fluorescence range is low. And if you had low input, then the fluorescence range on this axis is high. And what is showing you here is really the spread of the data points that you would get if you did many experiments. And this is showing you where 95% of all the data will reside. Okay, so that is a range in which this happens, and you can see there's quite a lot of variation that occurs, and that's because the measurements themselves are not that advanced yet that you can get those signals done very accurately. So these are the simulations, and based on the simulations, now you can look at various inputs here and look at the outputs there, and you look at the response curves which are formed, and this is very standard. So you will see that the reduction that you see, the inverse response that you see, will just depend on where your initial condition is, and how much is the spread over which you're looking at, how long. So this depends upon in this case what is this axis here that was shown independent of this, but you see here it is shown as IPTG, and that is the lag response that you see on the white yellow fluorescence protein, and then you can see based on different conditions you get different kinds of responses. Okay, and you can read this whole thing in that paper, like I said. Okay, now I'll just do a little bit of this because since we developed the topic, so I'll just do 1 or 2 slides more and then kind of conclude this topic, because we will be again doing this later on. Okay, now you see here, what is an inverter? Inverter means that if you have a low signal on this side, you get a high signal on the other side. So what it is showing you is that here there is already a gene that is getting synthesized. The promoter is already bound here and is giving you the synthesis on this side. So the output that is taking place it means that you don't need anything on this side to really get synthesis done on the other side. So that synthesis is really a high input output that's taking place. Now you can have the reverse case situation here. So you have mRNA already there, but this mRNA will go and be used to synthesize these input proteins, which are repressors. The repressor can bind on the site and then stop the synthesis of the gene or the transcription of the gene, right? So you can see that you have a high input here, but you get low output there. And this is the kind of symbol that is used. There are other ways of representation also, but generally we'll just follow one kind, and that should be fine. All right. Now, when this is the part of the modeling part. So the reason I'm showing you here, this is a very nice way that this has been depicted in terms of functions. Rather than telling you what the functions are or the specifics of how you're going to write out the model, and it's very easy to understand in that case. So what you have is an input function psi a that is operated upon. That gets into a function L, which is a translation function. So this will operate into that, and that will give you a function which is the input function for this cooperative binding here, or is the synthesis of the mRNA. Now, that cooperative binding will again have a certain kind of function, and that will be so this you could look at this. The input function here. I mean that it's an input that is known. The nature of the input is known, but this functionality here is slightly different from that functionality. So this means how does the output, which is phi a, and the input related? That relationship is shown by this L. Similarly, this rho a that is here and the phi a that is here is related by this transfer that takes place, this functionality which is here, which is the cooperative binding. Similarly, when you're looking at the output mRNA that is here, and look at the transcription that's taking place here, so then that is going to be shown by this translation function T. And you can see the nature of the functions that are there. So one after the other, you can see how the shape changes here. You have some kind of a delay, short delay. But here there is, of course, no delay here, but essentially that there could be a shift here later on. You can read about it where this whole curve shifts to the right. Then you have this phi z, which is here, which is going to be the inverse response, which takes place here, and then you can see that this will get modified because all of these together will give you this complete response that is there. So this operate into this, operate into this, gives you this response here. Okay, so that's how it changes. All right. We talked a little bit about this in the previous class, which is the pulse generating network. And what I told you to look at was, notice that there you have at this second promoter site, you have two signals which are there, which are opposing each other. One is an inhibiting signal, the other one is an activating signal. Whenever you have these opposing effects, you will get what is called a pulse response. This is going to be acting like a trigger. So this has been utilized as a motif throughout the biological world, and they appear as individual motifs maybe sometimes, or they'll occur in cascades, and the purpose is the same, that essentially there is a trigger, and this trigger, the duration, the magnitude of these triggers are already pre-matched in terms of parameters. In the parameter space, if I were to look at, it's as if I were to personify nature, then nature would have been sitting down somewhere and essentially working out all the details of this problem and saying, well, okay, this set of parameters works because now in this cascade, one after the other, I want this much of response right at the end, right? So if you were to pick up one organism and you go through this whole thing, I've said this before, and you pick up all the data, and if you can actually carry out one simulation, you'll find that you get a response which is something that you expect. The moment you tamper with the parameter space, huh?
I just small out here. So for the generation of the pulse,
Yeah.
do the repression and induction need to be coupled themselves as well, or can they be like completely independent?
They are not completely independent because there will be a time span over which they will have to couple, right? Otherwise, you won't get the response. Otherwise, what will happen is the repression will give you the inverse response, right?
Right.
And the activation will give you the upward response, right?
Right, sir.
If they don't overlap, then you will not cancel out some parts of it to give you the response, complete response. Do you get it? Maybe I'll draw it
So
out.
like,
Let me draw
for
it out.
example, if
Let me see if I can draw this out.
okay.
Can I do this? Oops. I always get this problem when I come to this page. Maybe I have to escape from here and I go to this. I have two touchpads from which I always get confused. And okay, so okay, now I can draw it right here. So see, if I were to look at this as an axis for the response, then if I were to look at the first part of it, the repression part of it, I'll get a response like that. And if I look at for the second part of it, the decouple, oh sorry, I'm on the wrong page. Yeah. Oops. Okay, anyway. I think you will be forgiving me for this drawing. So you'll have a repression that goes this way and an induction that goes the other way, right? And if you don't have them overlapping, you'll get them separately as I showed you upstairs, right? On this part here. If they are overlapping, then if the repression comes this way and you have the activation going this way, then this part cancels out, that part cancels out, and you get this response. Got it?
Okay. Yes.
That's the pulse. That net effect will be the pulse. So you could, so in principle you could write so if you're asking in terms of writing out the equations as separate, yes, you can do that separately. Okay, but
so
when
like
you write,
for example, if in this network, if S1 did not like affect R2,
right,
since S1 is inducing and R2 is repressing the final element.
Yeah, correct.
So if S1 did not like affect R2, then would like the situation be the same or would it be a bit different?
No, it would be different. You see, if they're not repressing, so I could then write out these two. Then what will happen is this Z equation that you see here, this one, sorry,
Yes.
this one. This would be a summation or some other logic will come here, right? Either
right, so
I could put a summation here, and I could have another K, maybe 1, I mean 2, 2, 1, maybe I don't know, 1, 2, right? So 1, 3, maybe I don't know. You can write it as 1, 3, and then you can put this out. So now what will happen is you would actually have this function and this function not multiplying onto each other, but giving you this kind of functionality where you can have this also. So this when you combine it like this, you're essentially saying that I'm not looking at the summations like that separately, but I'm giving you one function that is like that. Okay.
Okay.
Whereas if I were to separate them out, I would get this functionality which is here. Clear?
Okay. Fine. Sir.
All right. So there are ways to write it, different ways to write it. And if you look at the different papers or textbooks, then you find some more examples of this kind. Okay. All right. So let me just see if I can get the eraser here. No. Yeah, there there's the eraser. Oh, I'll leave that. I get so bothered with this. I'm not getting a hang of this. All right. So let me come back to this. So what you see here now is that there is another thing that happens. This is a concentration band detection. Now this French flag um, kind of a logic gate, as they call it, is predominantly found in developmental biology. Okay. The reason being that in this developmental biology, what you find is that you get cells getting expressed at certain locations in the developing embryo, right? And now it's a miracle, that there is this spatial temporal. You just think about it, how complex it can be, especially let's say if you look at any mammal, mammal embryo or mammal fetus, right? So that is getting developed. So and it starts from just single cell, two cells uniting, and then then that breaking down and kind of multiplying and getting you a mass of first set of cells that are there. And just based on signals that are there, it starts synthesizing certain proteins given at certain locus. And the timings are so important, because if the timings go incorrect, then you're going to get a mass of cells that are not going to be representing the animal itself, right? Now, exactly how it happens and how this has evolved is completely mind-boggling. But the fact remains, it's something like this, that again I have to maybe escape and go up here. So so if I were to if I were to look at a tissue, and let's say I'm looking at a center here, and you can see that in the slide there, also in the bottom slide, part of it can be seen. Then what happens is, in space, this is going to move. This is going to diffuse out this direction, right? Spatially. So if I'm looking at a tissue region as large as that, it's going to space it. So if I were to look at the concentration as it changes, then the concentration is going to go down like that, right? As it moves further away from this point where it is originating. And now, as it's originating, so you have cells along here. All these cells are there in this part of the tissue. Now, there are categories of cells which are here. And in this case, usually they talk about a French flag, means they usually break it up into three divisions. So we don't know if I don't know if people have found or discussed more complex motifs. But let's say we have three regions that are defined. Then there are some cells that are going to respond only in this region of the concentration. If I were to look at that region of the concentration, that much. Some cells will only respond at this region, and some cells will only respond at that region. And then depending upon that thing, and the duration, so the signal has to be there for a certain duration. If this signal of this, is there for a certain duration in this region, it's actually going to then synthesize, use this as a signal, and then taking that signal is going to synthesize further other compounds within those cells. Okay, and then those are going to grow, and then they would have locuses, and then they would give cues, and then this network starts developing. So starting from, you can imagine that what we're considering is only one set here, and as this expands, you'll have cascades of these all responding one after the other as the cells develop, right? And then they will build different kinds of tissues and differentiate in different kinds of cell types, and each of the cell types will grow. They know when to stop, right? For example, if the neuronal, the first, usually for a vertebrate, the neuronal, the spinal cord is the first thing that develops, and then the spinal cord folds inwards and then forms a brain. So if something goes wrong, then you keep on synthesizing something, some part of this tissue, then you will have either an overgrown brain or an overgrown, spinal cord. And in fact, some of the diseases that are there in children, young children, is, you have a neurofibroblastoma, which is there in children of age 1. They usually, some part of the kidney or somewhere, somewhere which is linked very close to, or, I don't know exactly what tissues, but I'm sure that I know of cases where the kidneys are the organs that are affected, and some part of it, they just didn't stop growing. I mean, some component, some cells within them, and obviously now that's going to look like it behaves like a cancer. So you have to have either it removed and treated. So if you look it up, it's really scary, and if something goes wrong, and it does, given the environmental condition that we live in, and with all of this pollution and other things that are there, things can just very easily go wrong. And it's a miracle that we still have so much of robustness in these circuits, that exactly when and how much concentration and what timing, within how much time things are going to work. It's all encoded in this kind of process, and it's amazing. So let's look at this down here, and let us look at the example that is given here. This is homoserine lactone, which is used as a signal. This is a quorum sensing, part of a quorum sensing, uh, uh, molecule. So you have a O63 HSL normally, but so these molecules are essentially excreted, and then so you can have cells which are on the outside reporting differently. So this has been used in this case to essentially give it as an illustration. So if you have one response, then you get one kind of signal. If you have multiple response, you get multiple signal, but you will essentially get these patterns which are there. So you'll get a pattern in the region that this will be responding to, depending upon the circuit that you've designed. Now, keep in mind that these circuits that are getting designed, if you were to do it artificially, then there is this problem of what we call kinetic tuning. There are two things that I have to do. The amplitude, the kinetic tuning means that the timing in which these things happen, and the amplitude that you get, as well as the diffusive tuning that is required. So how much distance shall it travel before you really allow this response not to have an effect on the cell types that are there under the influence. But of course, when we are doing this just for a study, all we are doing at this stage of our research on this area is only looking at the signals and saying, wow, oh, look at that, right? But if you look at how nature has evolved, and how these different motifs really contribute towards various kinds of cell types, it's amazing. So if you guys are looking at this, as an area of research, it's a wonderful area of research. This is where things are going to happen, and we can talk about some more things, later, maybe towards the end of the course. I'll tell you a little bit more of where all the energy is going towards, in this area of biological research now. Okay. So so this is showing you the bandwidth circuit that is there, and this slide I've taken from the this paper by Ron Wise and Basu. So essentially it's showing you these molecules coming in, the HSL molecules, and then you have two circuits which are there. So one is the X and the Y response that is there, and both of them give you this Z response, and the Z response then combines, and both of them, which means that both of them are inhibiting, and then that gives you the GFP, which is here, and that is going to respond in a certain distance, and you can see that response is going to be there, right? So this inversion that you see is going to take place here, and so you can see one signal is there, the other signal is going upwards there, and so in between, you get a response between that region where this response is going to take place. When both of them are together in a certain value, that's the response that happens. So at low value of this and high value of that, but not higher than that. So these are very, very nice circuits that are there, and you can go through all of those. When there are two ways to do it. I can send you the paper, you can look at it, and of course you can wait for the lecture to happen later.
Sir?
Yeah.
So the circuits which we are discussing are all these synthetic circuits, right? Like in all these we are saying that reporter genes are at the end. So
correct,
for the purpose
right?
of studying, we have
correct right now is doing, but these are all picked up from what you have to remember, that these circuits have been seen in actual cells, and they've been picked up from there, and then they've been studied in synthetic circumstances, so that you can kind of do all the variations and understand the entire thing completely, right? But of course, keep in mind that you're looking at one such logic gate or one such motif separately. Whereas in a cell, they are not going to happen like that. It's going to be happening in complete, tandem with the rest of the genetic machinery that is there in the cell. So for example, just think about it like this: that you have a cell, you have inoculated, and if you were to just observe, so when you inoculate cell in a shake flask, you have got 10 to the 6 cells are usually growing, and then you wait until it goes to 10 to the 8, and then you start working, doing the inoculum. But if you just look at the transversal of or traversal of or the trajectory that one single cell takes in its lifetime, you will find that grossly at the macroscopic level, the cell is growing. At some point, it's dividing, and then these daughter cells are again growing, and then at some point they are dividing. That's what you're going to observe, right? But if you look deeply, then you have to try to understand, when is it that it gets the signal to divide? So that's the first question. Then you go backwards, and then you are getting these nutrients. The cells are taking up these nutrients, and they're growing, and they're producing these hundreds and hundreds of genes, one after the other. And so again, at that level, all you're seeing is mRNA getting translated to proteins, and then they are doing the cellular function at the metabolic level. But what you're not looking at, and if you go deeper than that, then that's where the main thing comes in. So there are one or two cues from the environment that are coming into the cell, or maybe many more. It just depends on how you're designing the experiment, and they are coming inside the cell, and then inside the cell, it's hitting on one of these genes. These genes are getting synthesized, and as these genes are getting synthesized one after the other, they're going to have this circuitry that are going to interact with each other. And one of the small motifs is what you're doing here, one or two of them, right? One after the other. But just imagine hundreds of these circuitries which are there one after the other. Then how complex this whole thing becomes. Okay.
Yes, sir.
All right. Now let me come back here, and so this is a bistable network that is shown here. Essentially, you have a feedback, inductive, positive feedback that comes from R1. So you're synthesizing a protein that gives a positive feedback. More of this is synthesized, and then when this comes to P2, then that gives you a Z. So if R2 is not there, then so the condition of this switch is that it's going to be on or off, right? So there are two conditions at which they're going to stimulate. So what happens if you look at the synthesis, because of this feedback, it's going to jump to this state, and if you switch it off, it's going to come down this way, and then this condition, that when it comes in this condition, is going to see this low condition here, energetically low condition, is going to jump down, and then so you have up here and a down there. So you have this bisimilarity which is there. So the bisimilarity is very well understood like this. We have an inducer concentration with respect to the expression level, and so you have so you have this condition here. So you have this is called a saddle node. SN is a saddle node. Okay. So if you draw it on a phase portrait, then it will be like a hill, and so it can, if you look at the top of the hill, or an inverted hemisphere. At the top of the inverted hemisphere, if you put a marble, it can just travel downwards, right? It's not going to be stable. But exactly at the center, if you are able to put it there, it will be there. But the moment there is the slightest perturbation, it's going to go down, roll down, right? So that's what it means. So this one is right here. This one is also unstable. The stable condition is there for this one. Okay. All right. Now the toggle switch. So we have one more condition here, which is the toggle switch. So this one is going to be either on or off. So if you have, so look at this I2. If I2 is present, then it's going to really de-repress this one, which means that this gene is going to get synthesized. If this gene is going to get synthesized, that is going to give you P1, and P1 will then give you the gene R2. Then it comes back here, right? So this is so if you have I2, then I2 will give you an on. If you give an I1, then I1 will give you an off. So you can just keep on pressing it. So the first time it will go round, it will give you an on. The second time it goes round, it gives you an off. So the transcripting activator R2 binds to the promoter P2, and that inhibits the synthesis of so R2 will come here, and that will inhibit the synthesis of this. So the first time you saw it was going down this one, it was promoting the synthesis of R1. It goes on this one, it's going to switch off the synthesis of R2. Similarly, if you were to give I1 here, then likewise here it's going to de-repress this, synthesize this one, and then this will be then repressing this, you go back here, then that stops this one. So that's the way it goes. Okay. So read about this. This very nice and interesting one. And then we have also so this is actually a demonstration of that, really. So you can see that if you have IPTG given, and then there are different temperature conditions that are at which this happens. So you have the normalized GFP on this axis, and IPTG being added here, and gives you the time in hours as things happen. So if IPTG is there, it comes up here, then it goes down, right? So and because it is coming down here, then again it gets resynthesized because the other one will come up and give you this signal that is here. Okay. So you have IPTG and ATC. So you can see. So this first signal will give you this one. The second signal will also give you the depression. And if you look at both of them together, so this continues here, really, but at this point it goes down, and this goes down here. But you see when you have the TAC. So this is the TAC and the PKE 106. So this is an example of. Let me go back here. The first one. So this is the oops. This is the circuit diagram showing you this one, which is the same as this representation that is taken out from there, and it can show you that you can see here this part and this part, right? So it gives you this signal which is there. So the cell count. So essentially, what they do is they do a fax, and then they look at the cells which are there, which are giving you the signals. So you can easily see the separation that takes place in each of these cases. Okay. All right. Now there are some more complex circuitries that are there, and I'll not go into that. I'll just come back to it when I'll discuss that paper later. Right now, what we're going to do is we're going to look at oscillatory network. Okay. Now the oscillatory network that I talked to you about. No, I didn't talk to you about this, but I talked about this the student, right, Basu. So the first thing that they did was they looked at this repressolator. Okay. So they had designed this, and then they got it to working also. So look at the circuitry that is here. So R1 inhibits R2 synthesis, R2 inhibits R3 synthesis, and R3 inhibits R1 synthesis. The three inhibiting cycles one after the other which are there, right? And this is the model for it, where this I will be the value for the subscript that is here, and you could have different components that are there, which is this Rj. So it depends upon, because this J will be a different value, the subscript will be different. That's the reason why it's shown differently here. Okay. Now the repressolator will have a signal of this kind. It will have an oscillatory signal. And why would an oscillatory signal be important? Yes, somebody tell me. Anybody is listening or not?
Sir, we are listening.
Not sure.
What I'm asking is, Why would an oscillating signal be important in cells? One example. Nishantan is doing one for his term paper. So it is important. Yeah, circadian rhythm is one. Diurnal rhythms, for example, in plants, it would be very important. When the light would be light coming in, and the light that we see affects photosynthesis. So that becomes important. And the other thing that is important is it comes in important in cell cycles, right? So it has to know when the next cycle will happen. So that is also timed. So there are many cyclical processes within cells, and those cyclical processes are essentially taken care of by this kind of circuitry. Okay, but of course, this precipitator is not one of those. It's essentially picked up as a motive, and then just investigated to show that it's possible to have something in a cell. You can program it inside a cell. Okay. You also have mixed positive and negative feedbacks also, and this also will be again the same thing. If you have something like this, is going to act like a trigger. So you're going to have this pulse response coming in here. So that whenever both of them come together, there will be some kind of a trigger that's going to essentially synthesize more of this. Okay. If both of them come together, then there is a pulse. So there will be a duration over which this will respond, and then it will stop responding. Once. So initially there will be a feedback which is going to increase it, but when this starts coming in, this will go down. So this response that you see here is going to be a pulse that goes into the activation of this R2. And let's look at the last slide here. Oh, I didn't even mark it out for you. So this is the same one actually. So I wanted to put some things here, but I've not done that. So we'll talk about that later on. So essentially, I wanted to put in maybe some slides to show the responses which are not shown here. I'll do that later. Okay. So we stop here, and you can ask me questions on this topic. Yes.
Sir, I had a small question, sir. You talked about like IPTG. So, sir, IPTG is something which is used for like any kind of inducing any kind of promoter. Like we use that for lac operon.
Lac operon. So the LAC-I protein. So the LAC-I protein will be. So the lac operon is being used here in this case, but only part of it is not being used for the rest of the synthesis. But only that part which is required so that it actually controls some other gene which is upstream or downstream, sorry. Right?
Okay, sir. Okay, sir. Okay.
Any other question? All right then. So we stop here. Nishantan, in the last class you were on for nearly 3 hours after the recording. So I forgot to stop the recording and.
I'm sorry, sir.
No, not you. Ratnesh, sorry, not Nishantan. Ratnesh.
Yes, sir. I forgot to leave.
Okay, all right. So I'm going to stop the recording in that case.